Felix doesn't care about you. Isn't that
And
awful?
Lauren, soft sticks.
Hi.
Hi.
It was anticlimactic.
It was anticlimactic. I think it would have been better if I got to
I
do
think
it.
you would have done with more verve.
I think so.
Okay. Let's start where we started in the back room, which is how did you get involved and what was pushing, what was your driving force and the reason you got into science and then into research?
So when I was younger, I guess I was always interested in science, in how things worked, how we came to be. I think I was always interested in health and medicine as well. And so after high school, I chose to do a science degree at Melbourne Uni. And that's kind of, even though you obviously learn all of the basics in high school, that's when you really kind of break it down into the different fields of science, biology, chemistry, maths, physics. Quickly learnt that physics, maths weren't really for me. But what I really loved was biology. I loved learning about cells, all of the compartments in the cell, genetics, how we're kind of coded. And then...
Are we coded? How are we coded? Tell me.
Well, I guess I think thinking about patterns of inheritance and, how are we all different? And I think, obviously across humans, a lot of us are the same, but we're also very different, right? And I guess, okay, I'm getting a little bit lost.
It's a tricky question.
It is a very tricky question.
Shall we not answer it? And keep
We
on going
won't.
back
Yeah,
to
we'll
this?
keep going back.
Excuse me.
But I did find when I started my university degree, which would have been in 2011, next generation sequencing and our understanding of DNA had really kind of advanced. We'd had the sequencing of the whole human genome in the early 2000s, which, obviously happened well before I was at uni, but then, it just really took off from there. And
That was a game changer, wasn't it?
it was, absolutely. And then the advent of next generation sequencing, where this was no longer such a laborious process, where we could all of a sudden do this sequencing in high throughput, has enabled us to understand, human genetics even further, but in terms of cancer has really expanded our understanding of the drivers of cancer. And I was driven to cancer research, really, I think because of my interest in how the genetics of cancer and how we could potentially treat it by targeting genetics. I think I found that really interesting.
It is. It's fascinating. So that's what you did at university. You must have done a thesis.
So at university, I majored. As I said, I was interested in health and medicine. Like many other people, I kind of thought I would want to be a doctor as well. But I really became interested in science when I was in my third year, in research, in my third year at university. So I majored in...
Start that one again, just without correcting yourself.
Just hold that thought, just from the aircraft.
Yeah.
It's okay, here comes one now. My favourite sound and joke. That's what he would always say. Wait for the airplane. Oh, good, here comes one now.
I live in Waterloo, so this is like constantly what I hear.
Well, my son used to live in Newtown, and I used to live in Lilyfield.
I was going to say, I also spend a lot of time at the breweries in Marrickville, so cut that part.
Where were we?
Stand by.
Thesis.
I think I was... so at uni, I majored in pathology, so we learnt a lot about human health, different diseases, and we did a unit on oncology or cancer. And I was really just taken by cancer research, and I did a research project in my third year at university. And then I really thought, hey, this is kind of cool. I liked that I always loved writing as well as, I was really interested in science, but I loved English at high school as well. I loved to read, I loved to write. And I think the aspect that I really liked about research was that you could kind of combine all of those three. You spend a lot of your time getting to read everything that's been done before, really putting your research into context. Why are you doing what you're doing? Obviously performing the research yourself, but then being able to put that back into context in the field and really see how your work is potentially going to lead to progress, I guess. And so I decided to do an honours degree at the end of my bachelor's. And so I did that at oh,
Here comes one now.
Sorry.
Can you just turn
Did your
that down?
bachelor's? We'll remember where we were.
my bachelor's, yeah.
Do you see them flying through the blue sky? Was it Timlin? Did you have that app where you could actually spot the aircraft when they were coming?
I
Make
do.
it useful.
Is there an app?
We're
Yeah,
connected
so
now. Yes.
So what flight is that? And it tells you what flight's above you right now.
And it's on.
Filming outside would have been a nightmare.
If you know when they're coming.
What was that?
It says, oh, no, there's another plane in 4 minutes. Quick!
Which way?
Andy clock?
Like that?
Good more. Good low, too much. Oh, good low, thank
So even when you're expecting someone to land, you go, well, they've just
Oh,
landed.
they're here.
And the phone goes, yeah, we know where you're now crossing the southern runway and you will be going to your gate in 12 minutes.
There's no one coming. It's
you. Do you want to cut? Let's go.
Let's go. Irish said, oh, we've got houses in Noosa, you
Mark,
should come
Lauren
up.
2, soft sticks.
Chillish to say no. So brother at the beach.
Camera set.
No, I wasn't ready for you. I was talking about Noosa.
We can talk about this if you want. I've been there. I used to go surfing there. In my,
What's that? Yes, I think I saw it.
I remember being naked on a beach with my girlfriend. Someone walked along the beach.
too much information there.
No, it's really good. Anyway,
Thanks for that
we
image.
thought, we better put our clothes on. And they walked up and they said, we're here to sign a petition to stop development right on the foreshores. There was nothing on the beach. There was nothing built on the beach. We signed it. Don't know how good that did. Oh,
Thanks for that, Steve. You went to university and did.
I did.
A bachelor,
Yeah. So at the end of my Bachelor of Science, I decided to do an honours degree. And I wanted to do research, but I really wanted to do something that was quite patient focused. So I did my honours at the Murdoch Children's Research Institute in Melbourne, which is basically in the same building as the Royal Children's Hospital. So a lot of the research that was done there was very patient-centred, and the project that I worked on was using a technology called RNA sequencing to sequence childhood patients with acute lymphoblastic leukemia, or ALL, and this is the most common form of childhood cancer.
You know you're going to have to explain RNA sequencing, don't you? Do your best.
So essentially,
Just try to think about not saying so at the beginning.
oh, okay, yeah, okay. In biology, we think about the central dogma. So everything in your genes is coded by your DNA. So we have DNA, which is turned into RNA, which is then ultimately turned into protein. And protein is what we think about as the functional unit inside your cells, the thing that actually does things. And so when we do sequencing of your DNA, that gives us a lot of information, or if we're talking about the DNA of cancer cells, that gives us a lot of information about those cancer cells, but it doesn't necessarily tell us what is actually going on at the RNA or at the protein level. And so by doing the RNA, sequencing the RNA, we have a little bit more of an idea of what genes at the DNA are actually expressed.
Just hold that thought.
Yep. Qantas?
It is a Qantas, and it's circling Bethlehem.
Would you like to cut?
Yes.
I work
Mark.
Lauren 3, soft sticks.
So there was DNA, RNA, and proteins.
Yeah.
Thanks for that.
And one of the main drivers of ALL is something called a fusion gene. So this is when you have a gene A and gene B, and they would usually be expressed separately. But what happens with these fusion genes is that the genes are cut in, one place or another, and they are fused together.
How does that process happen? How does it get cut and how do they fuse?
I mean, it usually
It's a natural process.
it's a natural. Well, it's not supposed to happen, but essentially these breaks occur in your chromosomes.
Do you want to start that again? Yeah, so you were coughing on
So these, I said it again. Breaks occur in your chromosomes, and then your chromosomes contain genes, and so those genes get fused together, which then ultimately that's at the DNA, and then this is getting very complicated. I don't know. I feel like this is
Okay. I think you're doing quite well,
Okay.
actually, but you can describe it as a natural process.
okay.
But let's go back to how they break and fuse.
Okay.
But it doesn't need to be 100%. Just general. Help the audience out a little bit
Okay.
as to what you do. It doesn't need to be
Okay.
all the way, but maybe a little bit.
Okay. Can I just have a drink of water?
Yeah, absolutely. Do you want to think about it and come back?
Yeah. Can we
Yeah.
do something else and then come back to that?
Sure. Let's talk about the history of cancer research, from your point of view anyway. How much of cancer research that we know today, how recent is our knowledge of cancer, and to what extent? I think we mentioned before, it's like really all the major breakthroughs have been since World War II.
Yeah.
If you could walk us through that a little bit.
For me, it's interesting to think about the history of cancer because so much of it happened before my time in cancer research. A lot of the big discoveries, now that we understand cancer to be a genetic disease, have really happened since the sequencing of the human genome. Obviously, a lot happened before that point. But in the early 2000s, when we first sequenced the human genome, and then beyond that, when we started to sequence cancer genomes to really understand what are the drivers of cancer, so those changes in the DNA of normal cells that are making them cancer cells, what do they look like across the spectrum of cancers that we see in adults, children, in blood, in your brain, in other parts of your body, whether that's breast, prostate cancer. We have a much more in-depth understanding of what genetic changes are actually associated with cancer. So that's obviously been huge and has been what has really been the focus of my career around the genetics of cancer and how we can actually treat it better by understanding the genetics.
That's okay. We'll move on to Qantas again. We'll move on to what you're describing in the other room when how they wouldn't turn off. The cells undetectable and they won't turn off and they kept on going. But you can move into that if you like afterward. Just filling in the time with the Qantas.
Another 20 seconds. Hold that thought. Quickly.
Ready when you are.
In terms of what makes a cancer cell, we kind of understand what we term the hallmarks of cancer, those features that define a cancer cell. These are cells that can evade our body's normal mechanisms of detecting mistakes when these cells are replicated. So when cells replicate and grow and divide, the genetic material in those cells are copied. And this process is error prone, as many processes are. But our body normally has mechanisms to be able to detect those errors and kill off those cells before they can grow and expand. But often what happens in cancer is that those mechanisms are compromised. They will be changed or mutated, so the body can no longer detect those mistakes, and those cells continue to grow and survive. And that's what cancer is. It's uncontrolled growth of mutated cells, of non-normal cells.
Great. And what are the hallmarks of cancer? You've just covered. Go into greater detail about how many hallmarks were there?
I think there are 6.
Okay.
I can't remember them all off the top of my head.
Okay. You went into greater detail with the hiding, how they hide in plain sight, and so they can't be detected by the killer. I guess you would call them the killer cells. It's supposed to liquidate them, but don't.
Yep.
Could you explain that in your words? Because I won't be on the documentary.
I can't remember what I said in the other room.
That's all right.
Yeah.
You knew it.
Yeah.
Well, yeah, it doesn't need to be the same thing. Okay, right. So one of the hallmarks is cancer cells go and they multiply.
Yeah.
Right? Why?
Yeah.
How do they get away with it without being eradicated?
Well, they can evade your body's immune system. So normally you would have cells in your body that are trained to kill these abnormal cells. And also within a cell, you would have mechanisms that are activated when that genetic material is mutated. So these signals would then tell that cell to kill itself, essentially. So it's a form of cell suicide. And those are also compromised in cancer.
So they're just growing out of control.
Yeah.
Could you say that?
So they're growing out of control.
Okay. So tell me more about the research that you've been. Well, did we cover what attracted you to research and why you picked cancer? You just said you found it interesting. Was there any deeper reason?
I think it's natural curiosity, really. I mean, obviously, a lot of people are attracted to cancer research for their own personal experience. For me, like many people, I have family that have been touched by cancer or friends that have been touched by cancer. But I would say the predominant driver was scientific curiosity and obviously wanting to make a difference as well. I think being attracted to medicine and wanting to help people is one factor, but I felt like in research, this idea that it's those, really fundamental discoveries and that understanding of cancer biology that actually progresses the way that we treat patients.
What's the coolest thing you've done when you've been researching, or the coolest thing what you thought was the coolest thing maybe happened to you, or that you're aware of, even if it doesn't involve you?
The coolest thing?
Well, the thing that turned you on the most. So that's really cool.
I think it is probably our mission as a whole and the concept of personalised medicine, which is essentially what my research is entirely focused on. So we use sequencing technologies, as I've described, to understand the genetic changes that occur in cancers of individual patients. So this is what we do can I mention Children's Cancer Institute? So this is what we do at Children's Cancer Institute with the Zero Childhood Cancer program, which was originated here in 2017, where we first started sequencing the cancer genomes of patients from Sydney Children's Hospital. And this has now been expanded to a national program where, as of today, all patients that are diagnosed with cancer in Australia receive some form of genetic testing on their tumours. And the idea of this testing is to effectively identify that driver, as I mentioned, which is that kind of one change that has turned, or that we think has turned that cancer cell, that normal cell, into a cancer cell. And so then if we can target that cell specifically based on that change, that leads to better, more effective treatments, but also less toxic treatments, as opposed to what you'd be more used to hearing about with traditional methods of treating cancer, such as chemotherapy and radiotherapy.
Thank God. Excuse me. Is the point, start building, how many drivers are there? Is it infinite number of drivers, or do you think you've narrowed them down to 50 or 2,000 or?
There are hundreds, thousands of drivers. In terms of the ones that are actually targetable, there are far fewer. So some of the proteins, traditional ways that we target cancer cells are with what we call small molecule inhibitors or drugs. And so they can bind to these defective proteins, essentially. But because of the structure of certain proteins, not all of them, so not all of those cancer drivers can actually be targeted in that way. So that limits what we can do with targeted therapy. So there are some patients where we may know what the cancer driver is, but at the moment we can't necessarily do anything about it with these traditional methods of targeted therapies or drugging.
How often do you get a breakthrough on the drivers? The entire medical, not just you.
Yeah. So in terms of, when we analyse sequencing data from patients, so the genetics of their tumour, we are looking for things that you kind of know are drivers, those things that are established, and we can go, okay, that's what's driving this cancer. But we're also looking for, and they may be specific changes. So you've got your kind of genetic code. It may be one single change where an A is turned to a T that can be the change. It could be something like a fusion gene where one half of one gene is abnormally fused to one half of another gene, and that's a completely different driver. But these commonly affect certain genes. And so we've got that bucket of changes where we know that they're drivers, and then sometimes we identify changes where they're in those genes that have been associated with cancer, but it's a different change. It's not one we're used to seeing. And so we don't actually know whether that is driving the cancer or not. And so most of my research focuses on actually understanding what those changes do and whether they are causing cancer, number one. And number two, whether they can be potentially targeted with drugs that we have that are existing.
That's always good.
And then the third bucket is things that we have no idea about, things that we identify commonly in cancer, but we don't actually know how it's contributing to cancer. But then also sometimes we have changes that we can't explain at all.
You got an example of that, or?
Probably not. Probably not. I think probably the only other thing that I could say is that kind of fundamental knowledge that we have of the normal human genome. So alongside sequencing cancers, we also sequence normal healthy material or healthy tissue. to understand what is normal and what is not normal. So it's also that kind of baseline understanding of the changes that naturally occur in people but don't cause cancer that helps us narrow down what could possibly be causing cancer.
Just cut for a sec.
Lauren, 5.
So,
Would you like to run me through one of your typical experiments? Just explain what you do so I understand, in case we want to come back and film you doing it.
In my research, I mostly use cancer cell lines or cells. So most of my work is done in a plastic dish, which is obviously very different to what happens in the body in a cancer. But that's the fundamental kind of starting point where we look at how a genetic change influences a cell's behaviour. So when we identify in our analysis of patient tumour samples, when we identify a change that we think is one of these suspected drivers, we use that patient's genetic material, so their DNA or their RNA, to be able to copy that change, and then we use viruses to be able to express that change in our cells in a dish. And so that helps us create a model of that change. And so then we can use a range of different experiments to understand how that change influences that cell's behaviour.
So, step by step, you get an aberration in the cell structure, in the dish, and then you act on that. Or is that badly expressing myself? I feel it is. I think you can do better.
So, I've said so again, I'm so sorry. We copy that patient's change using a technique called PCR. That's probably getting too complex, but we copy it and we can use viruses to be able to infect our cells and incorporate that change into the genome, into the genetics of that cell that we're using. And then the question is, does that change affect that cell's behaviour? And this comes back to those features that make a cancer cell a cancer cell. Do those cells grow more quickly? Do they evade normal mechanisms by which we try to kill those cells? Do they change signalling within those cells? And so by signalling, I mean we have certain proteins within a cell that are markers of cell growth or signals for cells to survive that we can use to understand how that change is influencing the cell. And then most importantly, we want to understand whether those cells that express that change may be sensitive to targeted drugs.
All that thought?
I've got another question waiting. It's about you threw me with viruses. Because yeah, I was. We can still talk. This is not the interview, but it's like, was Ian Fraser? Was it that he was? Was it Ian Fraser that did the cervical cancer vaccine?
Yes, the Gardasil. Yeah.
And then he was saying that 20% of cancers are viral. He said, it's only 20%, but it's still 20%. But I didn't know, and I don't think most people will understand that there was virus involved in the causation of cancer, I guess. And you're using viruses in this aspect too. Could you explain it to me so I can understand? Because I fear I'm not getting it right.
In terms of how viruses cause cancer and the way that we use viruses, we really use viruses as a tool to be able to model genetic changes. So we can use the virus to infect cells to be able to change their DNA or to incorporate a change that we want to within those cells. So that's how we use viruses. We also, in addition to those viral techniques, this is still a viral-based technique, but one of the technologies that has really changed the way that we model these aberrations or mutations is CRISPR, which is basically a pair of genetic scissors that we've really derived from bacterial cells. So it's part of the bacterial cell's normal immune system, but we can use this technology to be able to alter the DNA in other cells, in human cells that we use in the lab. So this enables us to delete genes to be able to understand how they might be contributing to cancer. We can change the expression of genes, so turn them on and off. We can induce mutations, as I said, so we can, add a single base, a single letter into the genome. We can cause those fusion genes by inducing breaks, cutting DNA in the genome. So that technology as well has really changed how we can better model these variants in the tools that we have, in the cells that we have.
Is that fun for you? Experimenting with all those things and fun. What's the most excited you've been?
I think all of these technological advances are exciting. I think it expands what we know about cancer. In terms of my research and how we're looking to go forward, I think I mentioned already that we have kind of DNA, and then we have RNA, and then we have protein. And we kind of go for DNA and we go for RNA because in a way they are easier to get. Whereas with protein, it can be hard from patients to be able to sequence their protein or understand how their proteins are expressed. But based on what we know, it's that protein information that is going to give us the best idea of what's actually happening in a cancer cell. What genes are turned on, what genes are turned off, how can we target them?
That was really good. Well done.
Yeah.
You got any questions?
A little while ago you talked about the fact that your normal immune system, or a way that the immune system attacks cancer cells or any mutations. You said that that's compromised sometimes by certain things that happen, but you didn't say what things are.
The healthy camera departments.
Mutations. So what are those things that change?
I'm not sure that that's really my area of expertise. Like I don't think I'll be super comfortable answering that.
That's okay. Also remember that we'll probably show you everything, and if there's anything uncomfortable,
Okay.
we'll take it out.
Yeah.
So if you like,
I guess one thing that may be good to comment on is how we can alter and use the body's immune system, which is an emerging, one of these alternate therapeutic approaches rather than what I mostly focus on, which are these small molecules. As I said, we can target some proteins, but we can't target all. Another emerging way that it has been around for a while, but I think it's becoming a really big focal point of cancer therapy is immunotherapies, where we can use the body's own immune system. So we can alter your T cells, which are the ones that would normally bind to and kill foreign cells or abnormal cells. We can alter them to specifically target cancer cells.
Okay, but what causes the cancer cells or ordinary cells to mutate? I think you were hinting at that, but you never said what it might be.
Well, it's effectively just sporadic, right? So there can be genetic reasons as to why, and I think I maybe alluded to this, I can't remember if we were in there or in here, but there is an element and obviously this is coming from the context of childhood cancer, but there is an element of genetic predisposition. So some patients, some childhood cancer patients have mutations in their germline, so mutations that are inherited. They're in all their cells, not just in their cancer cells, that actually affect how their cells repair their own DNA. So that process, which normally regulates, as I mentioned, when your DNA is copied and it's not copied well, there are errors in that process. There would normally be mechanisms that help correct that process. Some patients have mutations in their cells that alter that repair process. So that can be one way that those changes are not repaired. But then also these changes just occur often sporadically. They just happen just by the nature of cells growing, dividing, replicating their DNA and having that be an error-prone process. And so it's when that, in combination with these kind of the inability for that to go detected, that you get cancer.
It's an imperfect system.
It is an imperfect system.
I've got a broad question. Stuart.
Yeah, sure. But when you answer that, look at me.
Yeah.
But Uncle Andrew will ask it.
It's probably a stupid question, but how close are we to curing cancer?
Which one?
Yeah,
Yeah.
I think that's the. I think we.
Well, let's say breast cancer, which is pretty common.
So I would say quite far. I'm not sure if we want to put that on there, but I think it does raise an important point that cancer, the more that we learn about cancer and especially about the genetics of cancer, we once thought of cancer as one disease, then we were able to break it down into breast cancer, prostate cancer, leukemia. Now, if we think about something like ALL, acute lymphoblastic leukemia, that I mentioned, there are over 20 subtypes of that disease, that one cancer, that are based on their genetic features of that cancer. So it's not even that we can break it down and treat breast cancer as one disease. There are diseases within a disease. So, and I think the further along we get in therapies and trying to treat cancer, almost the more questions are raised and how can we improve the way that we do treat it, because cancers are also very clever. When we go down the path of targeted therapies, we often find, so when you're targeting that one specific change that occurs in a cancer cells then mutate, they alter themselves to be able to evade that therapy. And so all of a sudden you get what will start out with one cancer cell where it's been able to mutate, it will then grow and be able to cause, ultimately relapse, for example, in a patient, or treatment resistance.
Because, sorry, Andrew, go to your point. I think we asked someone lung cancer, and I think the response was something like, well, it's a cancer of the lung, but they're all different.
Yeah.
And they're all different depending on your body.
They are. And I would say and I guess in terms of the cure, we are closer with some types of cancers than we are with others. In the case of leukemias, we do really, really well. So a child diagnosed with acute lymphoblastic leukemia has a 95% chance of survival.
Is that recent?
That's recent. And a lot of that has been the result of both targeted therapies, but also really just chemotherapy and understanding how specific genetic features are linked to response to treatment and are then linked to outcome. So we know with some patients, we may be able to identify a specific genetic change in their tumour where we can say, hey, we can't target that, but we know that that patient is ultimately going to do really poorly and may not respond to conventional chemotherapy. So we use a really high dose of chemotherapy to be able to treat that patient really intensely in the first place to hopefully avoid that relapse. And that kind of risk stratification in treatment has largely led to these dramatically improved figures in survival, which I think were about 20% in the 60s.
20% to 95%.
Yeah.
That's substantial improvement.
Yeah.
So is it a case of, sorry, I'm butting in there, Stu.
Yeah.
Is it a case of the more you know about cancer, the more you realise you don't know?
Absolutely.
You better say
Say
that.
So I've said so again. The more we sequence cancer genomes, the more we uncover. We identify a lot of changes that we know what they do, and we identify a lot of changes that we have no idea what they do. And based on, my experience and I guess my bias, I think that the path to better treatments and better understanding those genetic changes are going to be both more sequencing of patient samples. So just the more patients that we sequence, the more we are going to understand what changes actually are commonly recurring in cancer and what may potentially be contributing. But also, being able to advance our approaches into how we sequence tumours and potentially moving into phosphoproteomics or proteomics approaches to understand how proteins are actually activated and functioning in cancers. We also know that cancers are what we call heterogeneous. So not all, you may have, a bulk of a tumour and not all of those cells have the same genetic features. And so this can be something that also leads to treatment resistance or relapse, because you may have 80% of those cells that have this specific genetic change that you're targeting, but 20% of them don't. They may be driven by something else. And so when you're treating a patient, you may get rid of that 80% bulk population, but you've got that 20% that's still hanging around. And unless you can get that patient is ultimately going to relapse. So something else that we are moving more to now is what we call single cell sequencing. So we take a tumour and we sequence all of those cells in that tumour individually to see how similar or how different they are, and whether we may need, multiple lines of therapy to actually be able to treat that patient well.
So it becomes quite broad range.
Yeah.
Because
What cancer's will to live seems extraordinarily resilient.
Cancer?
Yeah.
Yeah. They are very clever. Cancers are very clever, and they grow and adapt and they change to be able to kind of evade whatever we throw at them, which is a constant challenge. And so, and I've said so again. Another reason.
Don't worry so much about the so, it's fine.
You can just delete that, right? Yeah, I lost my train of thought.
Yeah.
But
You were describing leukemia in the other room and all the...
Hang on.
Sorry guys, AC just come on.
Hooray!
And speeding up.
Lauren 6.
So, well,
You can move in the chair that way. That's right.
we're back. We talk about leukemia in the other room, and you were talking about the number of cells in children that's why they bruised easily. You were taking me through the entire, it was leukemia, wasn't it?
yeah, I guess that helps explain a little bit about what cancer is and how cancer kind of comes about in the first place. But in terms of leukemia, what leukemia is a essentially a block in differentiation. So all of your cells in your body, I think most people would be used to hearing about what a stem cell is. So you start from a stem cell, and then those cells will change into specific cell types that have specific roles in your body. And so when we think of the context of leukemia, these are your blood cells that often normally have immune functions. And instead of them actually differentiating or becoming those mature cell types that have those functions, there is a block, and they become this immature cell that essentially does nothing but just fills your blood system. And so you have effectively your immune system is compromised and you're just filled with these non-functioning cells.
All these bludgeoning cells going around doing nothing.
Doing nothing, yeah.
That was good. Okay. Jane, do you have any questions?
No, I'm good.
This is the last chance, Felix. It's your turn to dive into the realms of biology.
It seems that what you're talking about is that most people seem to think that it's external poisons and toxins and stress that cause cancer, but you're saying that in fact these could be variations just at the genetic level, and you're looking at battling cancer by turning on chromosomes on and off, by finding gene sequences that you can activate or disactivate to kill the drivers. Is that what it is? Is that where it's going?
In terms of the cause of cancer, there is still a lot that is unknown. We largely think of it as a sporadic disease, or something that just happens, as I said, kind of as the result of these errors that naturally occur in your cells. There are, of course, examples of environmental factors that lead to cancer. I mean, lung cancer and cigarette smoking is probably the best example, sun exposure and melanoma. In terms of radiation, obviously there's also some examples there. But largely speaking, and I think, kids are a great kind of example of this, where they haven't had these long-term environmental exposures that adults have, yet they still have cancer. And there may be a predisposition aspect to that. We're certainly learning more about that, where we're now sequencing both in childhood cancer, we're sequencing both their tumours, their normal DNA, but also the DNA of their parents to understand, how that DNA is inherited and what is potentially a risk factor, essentially. Are there changes in that child's DNA, in their normal cells, that actually predispose them to getting cancer in the first place?
Because people are dying more of cancer nowadays because we've cured the other things that used to kill them.
Absolutely. It is certainly a disease of aging. Cancer is a disease of aging. But as I said, we also see it in children as well. So there is obviously a lot of life years to be gained if we can kind of make some way on curing kids that are diagnosed with cancer, or young adults. Absolutely.
Certified then, are you actually doing research now where you can activate the genetics within a cell? You can change them, you can turn them on and off, you was talking about turning chromosomes on and off. That's really intriguing. Is that something that you're all doing now? Is that a way to cure it?
So in terms of applying those technologies, so here we're talking about CRISPR technologies, where we do have the capacity to delete genes, turn genes on and off, induce mutations or alterations to genes. And we largely use those tools at the moment to understand how cancers are functioning biologically. This is all the work that we do in the lab. In terms of turning those technologies into therapies for patients, that is a long way off. But it does help us actually understand what genetic changes may be essential for that cancer, and most importantly, identify what we actually need to target to eradicate the cancer.
So the holy grail in the future is going to be bespoke cancer treatment with a pill or with a vaccine or an injection of some sort. You've actually figured out what's happening in the body and you go, aha, this is the holy grail. This is like 50 years in the future or something.
I think there is a lot of work that does need to go into therapeutic development. At the moment, we do still have a gap in terms of understanding what the drivers are. We don't know what the drivers are for every child's cancer, but at least in the Zero Childhood Cancer program, where we are sequencing these patients' tumours to effectively give them a therapeutic recommendation, can we advise their treating doctor on what therapies may be potentially effective? For all of the patients that go through that program at the moment, over 70% of patients get a therapeutic recommendation. So there's something that we've identified in their cancer that can be potentially targeted with a drug. However, that obviously still leaves 30% where we don't know. But we also have a gap in therapies because we have a range of cancer drivers, genes, that we can't target. So we need to, as I kind of mentioned with the more traditional ways of therapeutic targeting, these small molecule inhibitors that can bind to specific proteins, they won't be effective for all proteins. Those drugs can't be developed for all proteins. So trying to advance how we can effectively target those drivers is still a gap and an area of research that's highly needed as well.
That must be frustrating because you know what it is. You can see what it's going to do. You know what it's going to do, but you can't stop it.
Yeah, absolutely. And so at the moment, all the best that we can do is think about more indirect ways of targeting that particular mutation. So that particular mutation may lead to a change or an activation of another protein that we can potentially target. So there are ways that we're trying to understand that, but it is still very limited, absolutely.
I'm very happy. Are you happy?
I think so. I don't know.
No, that seemed very clear.
That was,
It's intriguing.
yeah.
Do you want to cut that?
I think